First of all, I would like to thank the organizers for the opportunity to present our work on T-cell profiling in long COVID. This work has been led by Georgios Koutsodakis, a senior postdoc in my group at ISGlobal. Immune dysregulation, including altered CD4 and CD8 T-cell phenotypes, has been proposed as a potential contributor to the long COVID pathophysiology. And characterizing both global T-cell changes as well as SARS-CoV-2 specific responses is important to understand this condition. Our hypothesis is that long COVID is associated with distinct alterations in CD4 and CD8 T-cell compartments, including virus-specific populations, compared to recovered controls. So the objectives for this specific study were to profile CD4 and CD8 T-cell phenotypes using a high-dimensional spectral flow cytometry panel to identify immune perturbations, distinguishing long COVID from recovered participants, and to assess SARS-CoV-2 specific T-cell responses in both groups. So this is a cross-sectional prospective study. We selected a subset of 181 long COVID participants from the long COVID cat cohort that this morning Rebecca Santano presented from Catalonia. And we selected a set of 45 recovered controls from a healthcare workers cohort, also from the same area, from Catalonia. We used the weighting method for the matching, considering different covariates. The participants in the long COVID cat cohort, most of them were infected in 2020, and they were recruited into the cohort between November 2021 and April 2022. And we followed them up for 2 years and with 6 visits, in which we collected blood samples and self-field questionnaires. For this study that I will present, we used PBMCs from the first time point of the study. And at that time point, the participants had a median of 595 days since the onset of the persistent symptoms. Both groups were quite comparable. Most of the participants had first an infection, where very few had first a vaccination, COVID vaccine. The median age was 49 and 46 years old for each group. And 80% were females, and around 50% had comorbidities at baseline. The main difference were hospitalizations. In the long COVID group, there were around 20% of hospitalized participants during the acute COVID infection, whereas only 2% were hospitalized in the recovered group. They were comparable in number of vaccinations, infections, days since onset of symptoms, last vaccination or last infection. And we designed a flow cytometry panel to interrogate the differentiation, activation, exhaustion status of T-cell subsets, as well as their effector functions. I won't get into details for the sake of time, but just to mention that we had markers to define the memory subsets of T cells. Also, we included cytokine and chemokine receptors to identify different effector subsets, such as Th17, that is related to autoimmunity, Th1, that's more related to antiviral immunity, or Th2, that's more related to the humoral regulation. And as well as CXCR5 that allows us to identify follicular helper T cells that are in circulation. There was a set also markers of exhaustion inhibition, and we also had a set of activation markers that allow us to identify antigen-specific T cells upon in vitro stimulations with the spike peptides that will give us the T cell responses to vaccination and infection, and MN peptides that will provide information on the immunity induced by infection. And we used positive controls and a negative control to control for the spontaneous stimulation. So here we have the immune phenotyping results. We found that the long COVID participants had higher frequencies of conventional CD4 T cells, and specifically naive T cells, higher frequencies also of Th2, Th17, and circulating follicular helper cells with a Th2 phenotype, as well as higher frequencies of regulatory T cells. In contrast, they had lower frequencies of CD8 T cells, in particular effector memory CD8 T cells and terminally differentiated CD8 T cells. Those are in the last row. In multivariable logistic regression analysis, adjusting by potential confounders, CD4 T cells, total CD4 T cells were still associated with long COVID, as well as total CD8 T cells, effector memory, and TEMRA. When looking at antigen-specific T cells, we didn't find any differences between long COVID and recovered for spike or MN stimulations, but we found lower frequencies of MN Th1 specific cells in long COVID. This was using, well, I didn't say it, but we used different combinations of the activation markers depending on the T cell subset. So for CD4 T cells, we use any combination of the activation markers, whereas for CD8, we only use CD69 and CD137. Well, so we found lower frequencies of Th1 specific for MN, and then for the regulatory T cells, we found lower frequencies for both spike and MN. For CD8 T cells, total CD8 T cells, we didn't find any differences. However, there was a tendency to lower frequencies of cytotoxic CD8 T cells that we define as those that express the CD07A marker, that is a deregulation marker, and therefore it's expressed in those cells that are cytotoxic. In multivariable logistic regression analysis, only the Tregs specific for a spike were lower in long COVID. We also perform unsupervised analysis of the immunophenotyping data. Here we have the flowsome clusters that we identify, in total 16, and were defined by the expression of the different markers in the panel. So the colors are the expression of the different markers. And you can see in the U maps that each of these clusters has a different color, and the island on the left are CD4s, the island on the right are CD8 T cells. From those subsets, our clusters, we only find 3 that were significantly different in long COVID versus recovered participants. You can see the CD8 T cell subset, it's in the circle there. You can see that it's more intense in the recovered participants than long COVID. And the other 2 were very low frequency cells, and you really need a lot of imagination to see any differences here. But you can look at the box plots, and that is clearer. So one was CD8 T cells of effector memory, or TEMRA, and phenotypes that is consistent with the manual gating and the previous data I showed you. Then we also found a CD4 T cell subset of phenotype of central memory and Th2 that were increased in the long COVID participants compared to the recovered. And the third one was a follicular helper T cell subset with a Th1 profile. And frequencies were lower in long COVID than recovered participants. So in conclusion, long COVID is associated with broad and persistent dysregulation of the T cell compartment, extending beyond the SARS-CoV-2 specific responses more than 1.5 years after symptom onset. Specifically, we found skewed CD4 T cell composition, including circulating TFH and TH viruses that could affect the B cell compartment. We found diminished effector CD8 T cell subsets, indicating maybe a reduced cytotoxic capacity, although this is peripheral blood, so we never know if this is reflecting what's ongoing also in the tissues, or really the cells are there, are migrating to the tissues and exerting the effector functions. Well, we also found reduced SARS-CoV-2 specific regulatory T cells, although we found an overall expansion in long COVID. And then there are persistent abnormalities in non-SARS-CoV-2 specific T cell population, indicating systemic and not only antigen-specific alterations. So overall, our results point towards long-lasting and functionally relevant T cell imbalances in long COVID, which may contribute to inadequate immune control and may represent rational targets for immune monitoring and therapeutic interventions. And I would like to finalize thanking all study participants, also the Association of Long COVID in Catalonia, that they have been involved since the beginning of this study. Also all co-authors, my group at ISGlobal, and our collaborators at the Jordi Gol, Institut Català de la Salut, and of course the founders that made this work possible. Thank you for your attention.